Hello, pretty packed house. So really excited to talk about our experience at Broad Clinical Labs with the SBX Technology. So Broad Clinical Labs, we're kind of nested within Broad Institute. We sit at this really interesting intersection between genomic explorers, resource builders, and clinical translators. So basically, folks across the whole entire spectrum. So who's looking for answers and new technologies, those folks that are trying to build up large databases so that consortia have access to really like a plethora of data, and then how do we take that information and actually apply it to clinical problems? Here are a couple of examples of some of the folks that we've been working with over the years, and they run the gambit, and a lot of these names are familiar. And I'm going to mostly be focusing on the clinical translator side for the purpose of this talk. At Broad Institute, we also happen to be kind of uniquely suited to test out a bunch of different platforms. So we typically get the platforms before anybody else. So when they're in alpha release, groups will come to us, ask us to try it out, give some real-world experience. We have the ability to provide input in real time before the product gets launched, which is really cool. In addition, we also have quite a large set of sequencers that are being run at scale. So this is like our current production lab. We have a large set of NovaSeq X Pluses. We have a large set of Revios. We have some Ultima systems. And then we also have some mid-plex and smaller sequencers as well. So when Roche came to us, I guess this was in the fall, and we had the first conversation about SBX technology. We were really excited. So a couple of the things that jumped out, and Mark covered this in the very first slide. The first aspect is there's a lot of data that could get generated from the sequencer. So that's really, really attractive. In addition, supposedly high accuracy. At the point, we hadn't really tested anything, and the data that we were seeing was coming directly from Roche, so we had to take it with a grain of salt. And then the speed of the system was also really, really interesting. So being able to run and sequence genomes as quickly as we're able to do it on the sequencer, that actually unlocks quite a lot of potential. And one of the biggest places that we immediately thought that we could unlock potential is actually using this technology for rapid whole genome in the NICU. So today in the NICU, and Stephen Kingsmore gave a talk yesterday. He gave actually two separate talks. This figure on the right-hand side actually comes from one of Stephen's papers, and his group and Rady Children's has been pushing the envelope on rapid genomes for the last probably decade or so. And what they found, and this has been replicated in many other studies, is that there's a huge diagnostic rate in the NICU. And so when every single child in the NICU ends up with a genome, you actually find answers in greater than 50% of the cases. And even in the cases that you're not finding an answer for, there's actually information that gets transmitted that helps in the management of that child. And so that's really important for a couple of reasons. So the first reason is obviously you're improving the care of somebody that is super sick and they're young, really young children. So that's really important. The other benefit is when you start introducing genome into the NICU, you end up with those kids actually spend less time in the NICU. And so that means now there's all of a sudden more beds, these valuable beds in the NICU for other patients that are severely ill to come into the NICU. And then lastly, the really interesting thing is that the health economics of this makes a lot of sense from the hospital setting, the healthcare industry setting, because having an early diagnosis actually leads to shorter stays. Shorter stays means less money that the hospital is taking out of pocket to cover the cost of that patient's care. And it's also less money that insurers are paying. So overall, this is a really attractive target. It's been a really attractive target. It's gained a lot of interest recently, and it's starting to pick up and become routine care. And so when Roche first came to us, they had this assay, and it worked, and we were kind of looking at it, and it was able to sequence a set of genomes in a certain amount of time, which was very nice. We wanted to know how could we actually dial down each one of the steps. So as Mark said, there's a lot of levers. So you could decrease library prep time. You could decrease expandomer synthesis time. The sequencing itself is a big dial. So you could actually have shorter sequencing runs and maybe load less samples on, and so you could get it as fast as possible. And then actually, how do we get variant calling to be as expedited as possible? Each one of these steps together, there's a lot of opportunity to really shave off a lot of time and get this as fast as possible. So we got the instrument installed in January. So our colleagues actually had a little bit of a head start on us, and this was mainly because we were moving our facility from one building into another with all those sequencers. You could imagine how much time that was. So in January, after the holidays, we came back. We had the install of the system, and actually within like 1 day, we were generating data. And so this right here, I'm just showing some of the initial data that we ran on the sequencer. So everything I'm going to be showing was generated in our laboratory by our people. All the analysis was performed in our hands, and all the data came from us. And so, you know, the coverage is definitely higher than one needs for a genome, but this was kind of like the first shot. And so we were just getting our hands to get used to the system and actually start thinking of those levers and how do we dial them up and down. But overall, the F1 scores for SNVs and indels were extremely impressive. And so right away, we started thinking that, you know, maybe this promise that Roche came to us with, I don't know, 6 months earlier, actually has some reality to it, which is really exciting. When we start looking through all of the variants and we see how performance is across different variant types, interestingly, you know, this is a nanopore system, and as you can see here, homopolymer performance is fantastic across precision and recall. And so this is like really, really impressive to come out of this system with such good homopolymer accuracy. In addition, high GC, low GC, really good performance, really strong overall. Similar patterns for indels. I'm not going to go and kind of point out the differences, but in general, the performance and profile is really, really solid for these variants. And so benchmarking with HG001 through 4 it's great, it's good, everybody's got to do it. But we wanted to know, are we able to find causative variants in individuals that have been diagnosed with genetic conditions? And so we started testing some different variant types, so cases with different variant types. The first set of samples I'm going to be showing are coming from just Coriell cell lines. Most of these are actual patients that were impacted with conditions that would be typically diagnosed in the neonatal setting. So this first case is a patient has propionic acidemia. It's an inborn error of metabolism. These patients, actually in the first couple of days of life, if undiagnosed and untreated, even with treatment, they typically have like hyperammonemic episodes and they crash metabolically really early. And so these kids do come to the NICU. And so you could imagine just being able to have an answer for these patients could actually influence the care because dietary management is a part of the treatment for these patients. And so this patient has a single base deletion in the PCCA gene, and we were actually able to detect it without any issue. This patient is homozygous. This is part of a trio from Coriell, and so we do see it as heterozygous in both parents, and I'll show that data in a little bit. We wanted to look at performance across copy number variants, so we don't really just care about SNVs and indels. We also wanted to know how are we able to pick up copy number variants. This is a patient, also a Coriell cell line, Duchenne muscular dystrophy. This particular patient has a 28 kb duplication that actually like duplicates a couple of the exons in the DMD gene. It leads to Duchenne muscular dystrophy. Gene's also associated with Becker muscular dystrophy. We were able to pick this variant up without any issues using the platform. Going away from the standard traditional types of variants, we're also interested in STRs, so repeat expansions are a big interest for us. And so the first patient that we tested, so that once again, Coriell cell line, but we looked at a patient that had a full mutation in FMR1, so clinically affected with Fragile X. This patient, we were able to run and detect the variant in this case. So. These were not the only cases that we actually ran. We looked through a whole set of Coriell cell lines with different variant types. So you can see the first cluster are all small variants. They were all detected. The next set are all copy number variants, also detected. And then the last set are repeat expansions with full mutations. And so in each of these cases, the causative variant was identified. And so just thinking about the continuum, Coriell, great, but how does this actually work with patients? And so Broad Clinical Labs, we're CAP accredited, CLIA laboratory, also New York State accredited and licensed, and so we have patient samples in-house that have been diagnosed in our laboratory. And when we looked at those samples and we reran them, these are patients that have been consented to have additional testing performed with residual samples. You're able to see here that this set of samples, we were able to detect the causative variant that we actually reported in our laboratory for each one of these patients, which is really cool. It's okay to be able to do this and see the variant in a VCF file or an IGV, but that's not really how we analyze cases in the real world, in the clinical setting. So typically we take a phenotype-driven approach, and it goes into a tertiary analysis platform. This is an image from Fabric Genomics, Fabric Enterprise platform. And what you see is that this is that PCCA propionic acidemia trio, and the causative variant was able to be actually prioritized using the AI. And this was without any training, so we ran it as if it was a short-read genome, threw the data in without actually even telling Fabric, and it worked perfectly without any issue and was able to find the causative variant. And this is what it would have looked like if we took this through report. Just note this was not used for clinical purposes. We were just testing out to see how we could do it. So we got a little bit ambitious as we were going through this process, and we thought, we are probably going to be really fast. And when you start looking, there's actually been a couple of Guinness records. So Kingsmore had the Guinness record in 2018 for 19.5 hours. And this was using Illumina short-read sequencing. Later, he was able to cut it down, his group was, to 13.5 hours. Really, really impressive. In 2022, you and Ashley's group using ONT technology was able to actually get a genome sequenced in 5 hours and 2 minutes, which is crazy fast. And actually going from patient to diagnosis in a little bit over 7 hours. And so we actually saw this and thought it was a great opportunity. And just without any training or really much effort, we were able to get it down into 6 hours and 25 minutes. And so we've only had this instrument for about 6 weeks. And so really the potential is huge. And we're really excited to see how low we could go. So this was a work of a lot of people. Here are the main players that have been involved in this project for the last couple of months. And that's it. Thank you.